Welcome to video 9 of Genetics the Code Broken. In this video, we're going to be discussing mutations, the different types, and we're going to do a quick summary on one type of mutation that causes a disease, and then you're going to create a reporting class about another that you choose to research. So a mutation is any permanent change in DNA, and they can involve either changes to chromosomes or changes to single genes. When we talk about mutations of chromosomes, there are two types. There can either be rearrangements or changes in chromosome number. If we talk about rearrangements, we're talking about where parts of a chromosome are rearranged, therefore altering the sequence of DNA. And this might be through inversions, where a segment of a chromosome is reversed end to end. It might be through translocations, where chromosome segments change positions, or it might be through duplications, where portions of chromosomes are replicated, resulting in multiple copies. When we look at changes in chromosome number, we're talking about where cells can produce either too many or too few chromosomes compared to the number, and it's usually due to non-disjunction during meiosis. We have trisomy, where there's 3 copies of a chromosome, and we have polyploidy, where there's an extra whole set of chromosomes, and it's where duplicate chromosomes fail to separate in mitosis. We then compare to mutations of genes, and that's where we see either base substitution or a frame shift. When we talk about base substitution, we're talking about a single base being replaced by another base, causing a point mutation. So we have the example here where the code should read CAG, but instead there's been a point mutation where it reads now UAG in a hemoglobin gene, and instead of the CAG being produced, UAG is read, and UAG codes for stop codon, and this is what leads to some types of anemia. And then we can see a frame shift where there's a single base either deleted or inserted from a DNA sequence, and this means that there's an extra base or a loss of a base in the codon sequences. It results in coding for different amino acids, and it often results in a non-functional protein. So one example which we'll just quickly look at is cystic fibrosis, and that's where there's a single base deletion from one triplet on chromosome 7. So that is a type of gene mutation. So instead of the proper function, which is a membrane protein that functions as a chloride channel being produced, we get an impaired function, and that impaired function means that there is an impaired secretion of chloride, and this increases the viscosity of cellular fluids, resulting in cystic fibrosis. So that was a really quick recap of mutations. Make sure you're in class for the research activity, and make sure you tune in for video 10.